What do all of these animal pairs have in common? The Visayan warty pig and the domestic pig, the red wolf and the domestic dog, the Iberian lynx and the domestic cat. On the left, you have a critically endangered animal on the verge of extinction, with extremely low genetic variance in wild populations. And on the right, you have an animal that can donate oocytes to save them via interspecies somatic cell nuclear transfer. Whether we want to admit it or not, human-related activities are the most significant contributor to the dramatic reduction of animal species and biodiversity on this planet. Simple knowledge of reproductive biology and genetics tells us that when the number of individuals in a species reaches a critically low number, such as the riverine rabbit, which has 90% of mature individuals in only one subpopulation, that species may as well already be labeled extinct due to physiological abnormalities associated with inbreeding depression. With almost 1 in 4 animals at risk of extinction, reproductive technologies, specifically interspecies somatic cell nuclear transfer, could be the tool we use to reestablish diverse genetics in threatened populations. So what is interspecies somatic cell nuclear transfer, you may ask? To put it simply, ISCNT is essentially the same technology that was used to clone Dolly the sheep, but instead of being implanted in another sheep, the donor somatic cell, possibly a fibroblast from the tip of a tail, is transplanted into a recipient enucleated oocyte of a different species. But why another species? Why can't we just streamline this whole process and put the embryo in its own species, like Dolly? Well, harvesting the ovaries of critically endangered animals for the large amount of oocytes necessary for cloning doesn't sit well with most conservation biologists, which is why using somatic cells cryopreserved deep in the back of a Swedish biological resource bank, and not germ cells, is an attractive alternative. To continue in the description of the process, the resulting oocytes, now infused with foreign nuclear material, in this case from a critically endangered black-footed ferret, develop into viable embryos that are cultured in the hopes that they develop into blastocysts in vitro. These blastocysts are implanted into suitable, closely related surrogate females, in this case a domestic ferret, that carries the developing clone of the donor somatic cell. The surrogate female is often of the same species that donated the oocyte. As simple as this sounds, you can imagine that scientists run into a multitude of molecular problems. The small molecular details that are essential to successful interspecies somatic cell nuclear transfer make this endeavor quite a challenge. Species-specific embryology and reproductive biology, chromosomal differences, and mitochondrial and genomic DNA incompatibility of the nuclear material and the oocyte are all road bumps that need to be addressed in order to produce healthy cloned offspring. One of the biggest molecular issues with somatic cell nuclear transfer is the oocyte's failure to restore to totipotent state in the transplanted nucleus. This is called nuclear reprogramming. Nuclear reprogramming is essentially the resetting of the cell memory established during differentiation to result in a normal embryo. This process only happens successfully in a small amount of cloned embryos, resulting in a very low efficiency rate. Most embryos instead show epigenetic deregulation and inappropriate gene expression before implantation, post-implantation, and even after birth. Many scientists agree that improvement in nuclear reprogramming will come with more knowledge on the molecular mechanisms controlling pluripotency. In addition to nuclear reprogramming, incompatibilities between the oocyte mitochondrial DNA and the donated nuclear material also are a roadblock to successful interspecies somatic cell nuclear transfer. Necessary functions of the body require coordinated expression of both mitochondrial and nuclear genes, sometimes referred to as mitochondrial genomic crosstalk. The incompatibility of these genomes results in obvious apoptosis and abnormal blastocyst development. One avenue scientists suggest to mitigate the effects of this incompatibility is to insert cytoplasm from the nucleus donor, in addition to the nucleus, into the donor oocyte to create a heteroplasmic oocyte that has an environment some would say is more friendly to the foreign nucleus. In addition to reprogramming and incompatibility, somatic cell type, quality of cryopreserved samples, and tailoring of the embryo to be accepted by the uterus of a genetically different surrogate mom are all obstacles. Continued investment in research and well-designed interspecies somatic cell nuclear transfer experiments are essential for overcoming these hurdles. Better understanding of reproductive biology and the unique embryology of individual animals will be key. Interspecies somatic cell nuclear transfer may not be a good fit for each species due to a variety of factors. One endangered species, however, that proved to be a good candidate is the gray wolf. Specifically in the Republic of Korea, these animals are rarely found in the wild. Because of this, there is the possibility of depleted genetic variations in their populations. Dr. Min Kim of Seoul National University of South Korea described in her publication the success of producing two cloned wolves from the isolation of an adult ear fibroblast from a female gray wolf and microinjecting the nuclear material into an enucleated canine oocyte. Within Dr. Kim's materials and methods, it is described that two pulses of electric stimulation were used during the fusion of the donor cell and ooplast. Selected cloned embryos were surgically transferred into the oviducts of the canine mothers. Only 2 pregnancies occurred, and in both dogs, 2 fetal sacs were confirmed. However, only 2 cloned wolves were delivered. Microsatellite analysis of 19 loci comparing the genomic DNA from the donor wolf, the cloned wolves, and the surrogate canines was performed to eventually confirm that the cloned wolves were indeed genetically identical to the donor wolf. Dr. Kim's experiment was not the first to successfully accomplish interspecies somatic cell nuclear transfer, but her results did show promise of using the reproductive technology with endangered wildlife. Like many hot-button issues within biotechnology, genetic restoration and interspecies somatic cell nuclear transfer in critically endangered species is no different. Reproductive biology and emerging technologies are not widely accepted and poorly understood by professionals in the field of conservation biology. Many conservation biologists fear, and rightfully so, that high-tech breeding methods divert attention and funds from protecting the foundation of the species, its habitat, what we as humans have essentially destroyed across the globe. In some cases, however, such as with the gray wolf, reproductive technologies may be the only opportunity for an endangered species' survival. Conservation efforts and reducing negative human influences should be first and foremost, but backup plans like interspecies somatic cell nuclear transfer, a potentially powerful tool in population genetics and in endangered species conservation, is never a bad idea.